Attorneys for Tyler Robinson tried to cast doubt on DNA evidence presented in court today during the second day of his preliminary hearing.
Robinson is charged with murder in the shooting death of political activist Charlie Kirk at Utah Valley University last year.
For most people, DNA is the closest thing we have to a witness that cannot lie. It doesn't forget. It doesn't get scared on the stand. It doesn't pick the wrong face out of a lineup at 2 in the morning. It has no motive, no grudge, no reason to shade the truth one way or the other. When a forensic analyst stands up in a courtroom and says a sample matches, most people in that room, and most people watching from home, quietly conclude that the case is essentially over. That's the story we've been told for 30 years, in real courtrooms and in every crime drama ever filmed. A swab, a match, a conviction. Roll credits. It is one of the most powerful ideas in modern justice, and it is far shakier than almost anyone outside a forensic laboratory understands. Then, over 5 days this July, in a 4th District courtroom in Provo, Utah, that comfortable story ran headfirst into a problem it could not talk its way around. Because a bolt-action rifle, with a single spent round still in it, was found wrapped inside a towel in a wooded area near where Charlie Kirk was killed. And when the DNA on that towel was tested and retested, it did not produce the clean, single, unmistakable answer the public imagines DNA always gives. It produced something far messier, something more human, something that 2 sets of trained, credentialed experts looked at carefully and walked away from with opposite conclusions. Before we go one step further, one thing has to be said plainly, and it stays true for every second of this video. Tyler Robinson has not entered a plea. A judge, not this channel, and not the comment section, will decide whether this case advances to trial. We are not here to tell you whether he is guilty or whether he is innocent. That is not our role, and honestly, it is not our right. We are here to explain something narrower and, in a way, far stranger. Why the single most trusted category of evidence on the entire planet became the exact thing that everyone in that room ended up fighting over. Because the answer, it turns out, has almost nothing to do with one man in a courtroom, and almost everything to do with what DNA can and cannot actually prove. To understand why the experts split, you first have to unlearn something. You have to set down the version of DNA evidence you've absorbed from 1,000 hours of television and look at what it really is. The witness we invented. Here's the version most of us carry around in our heads without ever examining it. A person touches something. In touching it, they leave their DNA behind. A machine reads that DNA like a supermarket scanner reads a barcode. And the result is binary. It either matches you or it doesn't. Clean. Absolute, certain. A yes or a no, delivered by a machine that has no opinion. Almost none of that is how it actually works anymore. And to see why, you have to go back to where forensic DNA began, because the tool that walked into that Utah courtroom is a very different animal from the one that was born in the 1980s. When DNA fingerprinting first arrived in criminal justice, it was genuinely revolutionary, and the early cases were built on something the analysts had in abundance: biological material, a pool of blood, a clump of hair with the root still attached, rich, generous samples packed with millions of cells, all of them belonging to one person. In that world, the barcode model almost held up. When you have that much clean material from a single source, the profile you pull is strong, complete, and very hard to argue with. Those early cases put serious criminals away, and just as importantly, they began pulling innocent people back out of prison. The Innocence Project has used DNA to overturn hundreds of wrongful convictions. That is not a small thing. That is one of the great forensic achievements of the modern era, and nothing in this video takes it away. But the science did not stand still. It got hungrier, and it got more sensitive. Analysts pushed the technology further and further until they could pull a genetic profile not from a pool of blood, but from almost nothing at all. From the invisible residue a person leaves just by touching an object for a moment. A doorknob, a steering wheel, a towel. They call it touch DNA. And it changed everything. including the certainty. The problem hiding inside the breakthrough one. The DNA at the center of the Robinson hearing wasn't a generous, single-source sample. It was trace material, the microscopic scattering of cells a human being can shed just by handling something. We are talking, sometimes, about a few dozen cells, occasionally fewer, an amount so small it is invisible, weightless, and until quite recently, completely untestable. And that tiny quantity is exactly where the confidence begins to crack. For decades, touch DNA simply couldn't be done. The machines weren't sensitive enough to read that little material. Then the technology crossed a threshold. It became sensitive enough to build a profile from a whisper of biological material where before there had been nothing. And here is the part that the field understood immediately, but the public never really absorbed. That same sensitivity, the very thing that turned touch DNA into a breakthrough, is also precisely what opened the door to the argument that unfolded in Provo over 5 days. Because when you are working at the absolute edge of what a machine can detect, 3 problems appear that the crime dramas never, ever mention. And all 3 of them showed up in that courtroom. Problem 1. The mixture when investigators tested the towel, wrapped around the rifle, the DNA on it did not belong to one person. It never resolved into a single, clean, one-owner profile. Instead, according to testimony from the State Bureau of Investigation, it pointed to 2 people. One was described as Robinson's roommate, Lance Twiggs. The other was described, and listen to the exact language, because the language is the whole point, as very likely Robinson. Sit with that phrase for a moment. Very likely. Not is, not definitively matches, not belongs to. The prosecution's own expert, testifying for the side trying to advance the case, framed it as a probability. And that wasn't hedging or weakness. That is simply what the honest science permits when you're dealing with a mixed, trace-level sample. When the genetic material from 2 or more people is tangled together on a single object, in tiny amounts, separating it cleanly and assigning each fragment to a specific human being becomes an exercise in statistics, not certainty. The FBI analyst who worked the sample, Amanda Backer, testified that after Robinson's roommate provided a reference sample for comparison, she was able to rerun her tests and attribute the DNA to 2 people. Think about what that sequence tells you. The interpretation of who was on that towel depended, in part, on having a second known person's DNA to compare against and, in effect, subtract out. The answer wasn't simply sitting there in the sample, obvious and self-evident. It had to be worked toward. That is normal, accepted forensic practice, but it is a very long way from a barcode. Problem 2. The partial profile, a clean, single-source DNA sample, hands you the full genetic picture, the complete set of markers that, taken together, can point to one person out of billions. A trace sample often hands you a fragment, pieces of the code, with gaps and holes where there simply wasn't enough material to read a given location reliably. And a fragment behaves very differently from a full profile. A fragment can be consistent with a particular person without being unique to that person. It can fail to exclude someone without positively singling them out. The difference between "this could be you" and "this is definitely you and no one else on Earth" is enormous when a life hangs on the answer, and trace samples very often live in that first, weaker category. This is the exact seam the defense pried at. Attorney Michael Burt pressed the FBI's analyst hard about the methods and techniques she used, and he distilled his entire challenge down to a single sentence. She can't match Robinson to the question samples. Read that carefully, because it is narrower and more precise than it first sounds. Burt did not stand up and declare that his client was somewhere else, or that another person is responsible. He didn't offer a competing story at all, and legally, he didn't have to. He made a far more clinical point, that the sample itself may not be clean enough, complete enough, or singular enough to point to one and only one human being in the world. It is an argument about the limits of the evidence, not a claim about the innocence of a man. And scientifically, That kind of argument is much harder to wave away than a simple denial. Problem 3. The transfer, and then there is the third problem, the one that genuinely unsettles the people who study this for a living. It is called secondary transfer, and it is the part that should make you rethink what the phrase, "your DNA was on it," even means. Here is the uncomfortable truth that the forensic field has known for years, and that the public has mostly never been told. Your DNA can end up on an object you have never touched in your life. It sounds impossible. It isn't. You shake someone's hand, and some of your skin cells transfer to their palm. They then grip a doorknob, a railing, a tool, and now your genetic material is sitting on a surface you never went near. You share a towel, a couch, a car with another person, and your cells travel with theirs to places you were never present. DNA is not glued to its owner. It moves. It hitchhikes. It travels through intermediaries, riding from person to object to person to object. This isn't speculation, and it isn't a defense attorney fairy tale. Researchers have demonstrated it under controlled laboratory conditions. In one well-known type of experiment, scientists had people shake hands and then handle knives, and afterward, DNA from a person who had never touched the knife at all was recovered from the handle. In some trials, that transferred DNA showed up as a major component of the profile, even though that person's hands had never made contact with the weapon. Now, the honest caveat, because this cuts both ways, and fairness demands it. Secondary transfer does not happen every single time, and it does not happen uniformly. How much DNA transfers, and whether it survives on a surface, depends on an entire tangle of variables. How much a person naturally sheds, whether their hands were wet or dry, the type of material involved, how long ago the contact happened. It is not a magic eraser that explains away all DNA evidence everywhere. Prosecutors are right to point out that its presence in a lab study doesn't automatically mean it happened in a given real-world case. But here is the thing that changed the ground under everyone's feet. It happens often enough, and reliably enough, that the two sentences, "his DNA was present on this object," and "he handled this object," are no longer automatically the same sentence. And relevant to this specific case, remember one detail already established in testimony. The two profiles on that towel belong to two people who live together. Roommates, people who share a household, and everything in it, which is precisely the kind of environment, shared space, shared objects, constant everyday contact, where the science says transferred DNA is most likely to appear. That doesn't prove transfer happened here. Nobody can claim that, but it is exactly why a defense attorney is entitled to raise the question, and exactly why the prosecution can't simply dismiss it out of hand. This is the quiet reckoning sitting at the heart of the entire hearing. For a full generation, a DNA match on an object was treated, in the public mind and often in the courtroom, as proof of contact, and contact was treated as proof of presence. The science has quietly, steadily complicated every single link in that chain. Match no longer cleanly means contact. Contact no longer cleanly means presence. And almost nobody outside the forensic world ever got the memo that the chain had weakened. How the same evidence splits two honest sides tibes. Now watch how one identical piece of evidence pulls two entirely reasonable, entirely professional sides in opposite directions. Because this is the part people find hardest to accept, that both sides can be arguing in good faith. The prosecution's position is not weak, and it would be deeply unfair to pretend otherwise. Their argument was never that the DNA stands alone as a lonely, load-bearing pillar. Chief Deputy Utah County Attorney Chad Grunander told the court, after 4 days of testimony, that the evidence was overwhelming and, in his words, devastating. But crucially, that assessment rested on the DNA sitting inside a much larger picture, one that prosecutors built out across 5 days of witnesses. To the prosecution, the trace on the towel is not a single fragile thread that the whole case dangles from. It is one strand woven into a much thicker rope, alongside surveillance video, witness testimony, and other evidence they presented. Pull one strand and the rope may still hold. They also made a purely procedural point that is legally sound. A preliminary hearing is not a trial. Its job is narrow, to determine whether there is enough evidence to proceed at all. And the fine methodological battles over DNA interpretation, they argued, belong in front of a jury later. Deputy Utah County Attorney Ryan McBride made exactly that point, telling the court the reliability of the DNA testing could be fully examined if and when the case goes to trial. The defense's position is also not a stunt, and it would be equally unfair to treat it as one. Their argument is narrower, more surgical, and in its own way more clinical. They are not obligated to hand the court an alternative version of events, and they pointedly did not. They didn't name another suspect or spin a counter-narrative. They did something more disciplined. They took the DNA evidence, held it up to the light, and asked the precise question the science itself invites. If this sample is a partial, mixed, trace-level profile, described by the prosecution's own expert as very likely, rather than certain, pulled from an object shared by two people who live together, then how much weight can that single microscopic trace really bear, when the stakes are a man's life? Because prosecutors have made clear, they intend to seek the death penalty. When that is the stake, the defense argues, the margin between very likely and certain stops being an academic footnote and becomes the whole game. Both things can be true at the very same time. The evidence can be genuinely serious and genuinely contestable. It can be strong enough to help send a case forward, and honest enough to admit it deals in probabilities. Those are not contradictions. They are the actual texture of trace DNA forensics, and the public almost never sees it laid out this bluntly. And that, in the end, is why the experts are divided. Not because one side is lying and the other is telling the truth. Not because one is competent and the other is a fraud. But because trace DNA lives in a zone the public has essentially never been shown, a genuine gray zone of probabilities, partial answers, statistical weighting, and transfer effects, where two careful, credentialed, honest scientists can study the exact same data and arrive at the boundary of what it can prove from completely opposite directions. The division isn't a scandal. The division is the science being honest about itself, in public, for once. Zoom out. This is bigger than one courtroom now, Kay. Now step all the way back, because what happened in Provo is far larger than one case in one state. Touch DNA is used in an enormous number of criminal investigations, all over the world, every single day. It has helped free people who were wrongly convicted and had already lost years of their lives. It has helped identify perpetrators who were certain they'd left nothing of themselves behind. It is, without exaggeration, one of the genuine forensic breakthroughs of our lifetime, and this video is not an argument against it. Used carefully, understood honestly, it is a tool of real justice. But its power grew faster than the public's understanding of its limits, and that gap is dangerous. Most jurors, most voters, most people scrolling past a headline still carry the old barcode model around in their heads. Match equals guilt, sample equals presence, machine equals truth. And the actual science has quietly moved somewhere far more careful, far more probabilistic, and far more humble than that mental model allows. When a member of a jury hears his DNA was on the object, many of them hear a door slamming shut, when the science may only be offering a door left slightly ajar. That mismatch is not harmless. It cuts in both directions. It can help convict a person on evidence weaker than the jury believes it to be. And it can also let a guilty person walk, when a jury overcorrects and distrusts DNA that was, in fact, solid. Neither error serves justice. Both flow from the same root, a public that was handed a breakthrough tool without ever being told where its edges are. Which is what makes this hearing quietly important, whatever the judge ultimately decides about whether the case proceeds. It dragged a private, technical, deeply nerdy debate, one that forensic scientists have been having among themselves for years, out of the shadows and into open court, in front of an entire watching country. For a few days, the public got to see the thing the experts already knew, that the most trusted witness in the modern courtroom does not actually speak in the language of certainty. It speaks in the language of probability. And most people had no idea. The question this forces is not, did this one man do this one thing? That is for a trial, for a jury, for a process with rules and protections, and a burden of proof, and emphatically not for a video or a comment section. That question is out of our hands, and it should be. But underneath it sits a second question, older, larger, and one that will still be standing long after this particular case is closed. How much certainty should we ever build on a few invisible cells, when the most trusted witness in the modern courtroom turns out to speak in very likely instead of beyond doubt? How sure is sure enough? That is the real thing the experts are divided over. It always was. This case just happened to be the moment the argument spilled into the open. And I want your answer to it, down in the comments. If a piece of evidence is powerful enough to help decide whether a person lives or dies, but honest enough to admit that it deals in very likely rather than certain, should that evidence ever be enough to stand on its own? Or does the entire weight always have to be carried by everything built around it? The video, the witnesses, the full picture, with the DNA as just one strand in the rope and never the rope itself. Tell me where you land and tell me why. And if you want more stories where the science quietly overturns something we all assumed was settled and certain, subscribe and stay right here with us. Because the closer you look at the tools we trust most, the more you find they've been speaking in probabilities the whole time, and we were the ones who chose to hear certainty.